Looks as if we've got everybody logging in now. So welcome to Fluidigm's presentation this morning. So we have Gloria Martus from the Field Application Specialist team here at Fluidigm, who's joining us today. And she is going to be speaking to you about implementing Cytometry technology for standardized high-dimensional assays for clinical research. You will notice that on the right-hand side of your screen, there is a comment box. Please type any questions or comments in here about the presentation. We're going to have time for a Q&A session at the end of the presentation, so we'll get to those then. So Gloria, I'm going to hand over to you and you can start the presentation.
Hi everyone, my name is Gloria Martus. I'm the FIS for Proteomics South Europe at Fluidigm, and I'm going to explain you today implementing the Cytometry technology for standardized high-dimensional assays for clinical research and how we're going to be able to analyze from cell types to tissue more than 50 functional and phenotypical markers at the same time on the Helios and Hyperion imaging system. So to start with that, like at Fluidigm, we have a complete multi-omics workflow, which spans not only from like what I'm going to explain you, which is mass cytometry and imaging mass cytometry, but we also have other solutions which will allow you to have this automated sample preparation for both for flow and mass cytometry, such as the flow conductor, but also it will allow you to have this automatic RNA-seq library preparation or biomarker discovery. So we do have our solutions for genomics, such as the single-cell genomics or the C1, or which will allow you to have this complete analysis of the cellular function and engineering with cell-to-cell interaction. Nonetheless, today, what we're going to do is focus on have our solutions for genomics, such as the single-cell genomics or the C1, or which will allow you to have this complete analysis of the cellular function and engineering with cell-to-cell interaction. Nonetheless, today, what we're going to do is focus on Cytometry technology. So we here is the Heliosystem. So this is the machine that will allow you to analyze more than 40 markers without any compensation, which is actually something that's very powerful because you will be able to obtain from one single tube all this information. And it will also allow you to have this comprehensive idea of what will happen in your cell without any compromise. You will not have to compromise any of your markers or any of your different cell types, because in one single tube, we will be able to measure all of that. It's a very proven technology because it has been chosen for a lot of different clinical trials and also used in basic research. I will explain you a little bit more into detail later on. We also have the Hyperion System, which is this big block machine that we put in front of the Helios, as you can see here in the image. And it allows you to visualize tissue architecture in FFPE tissue samples or even frozen samples with single cell resolution, even subcellular resolution, because we're going at 1 micron resolution, and we can actually measure more than 35 markers. So just to start, just like as a first introduction, it's a proven technology because we do have more than 1,000 publications until December 2020. And as you can see here, the ramp publication has gone up quite exponentially. And out of these publications, it's not only publications, but we do have more than 120 clinical trials that had used Cytometry technology. Some of them already have finished. Some are already new trials. So in 2020, there were quite a lot new ones using it in vaccine development, as we can see here. So 40% of our trials are in immuno-oncology, but we do have quite a lot of them in infection diseases and vaccinations. So you might very well know how flow cytometry works. This is just like a small summary. So we have antibodies that are linked to fluorochromes. Flow cytometry has helped us enormously in the last years to kind of measure everything we need it for, but it does have some limitations. One of the limitations is that just due to the physical nature of fluorochromes, there is a fluorescence spillover. So when we excite the fluorochrome with the laser, the signal that we will get out that it will emit, it will not be perfectly accurate, but it would just have this kind of spillover signals that would just get one into the other. There are some ideas of how, some strategies to avoid that, but still it's not perfect. And hence, this is why you need to do compensation. You will need to use a lot of tubes for compensation on that. Moreover, the fluorochromes are not stable. So to say the intensities that you will measure on a fluorochrome on PE, let's say here, I'm based in Barcelona, let's say in a machine here in Barcelona, will not be the same intensity that you will measure in a machine in Paris or in London or in Athens. So they have this highly variable signal intensities. Moreover, biological cells, so cells do like to shine. So we do have this background signal that comes out of the excitation of the cells just due to the laser excitation. So there is this emission of light that comes out of the cells. And this emission of light can be very narrow or can be extremely broad. And depending on the different fluorochromes you're looking at, the resolution of your signal will be very low. So to overcome that, mass cytometry is using exactly the same antibodies that you will use for flow cytometry, very compatible protocols that you will use in flow cytometry, but instead of using fluorochromes, we're using metals. These metals, this table, you might recognize it from your time when you were like at chemistry. And this table, we highlighted the metals that we're using in pink. We span from the 89 yttrium to the 209 bismuth. And the metals, they share like this common properties. They are non-rare, so they're easy to extract. They're non-biological, so we do not have any background. They're non-radioactive because otherwise they won't be safe. And they're high pure, so they are more than 96% purity. So this is how our signals look like. We are measuring from the 89 to the 209 bismuth, and we do have all these metal families. The isotopes are all classified depending on their mass, and they have these separate and distinct signals up to 1 Dalton resolution, which allow us to distinguish one from the other. There is less variable signal intensity. Why? Because all our machines are normalized every day. We have this tuning, and also the machines are normalized, and we run this EQ bits, so we run beats together with the samples, so we're able to actually compare sample day to day, machine to machine. And we do not have any autofluorescence because we are not measuring fluorescence. We don't care. We don't have any background because we do not measure any of the metals that might be present or the atoms that might be present in a biological cell, such as carbon, oxygen, hydrogen, iron, whatever you think. So right now we have 135 independent detection channels, which can measure more than 40 cell parameters at the same time without compensation or controls. One thing that is actually something that we can implement nicely in mass cytometry is cell barcoding. We're able to multiplex because we have so many that we can choose, and we don't have any issues with spillover. We're able to multiplex different samples in one single tube, and we have different solutions for that. So this allows us to have this improved data consistency, which shows that we have lower CVs in our signals and a better doublet removal, and mitigates also the cell loss because we will have a bigger pellet, and we also have like a higher workflow efficiency because it will save your time, not only for your staining, but also for the acquisition of the samples, and will save you money because you will use less reagents. So right now we do have 3 different solutions for that. So we have the Palladium-Based Barcoding, which is our first solution, where you use like 6 different stable palladium isotopes that will allow you to do barcoding up to 20 different samples in one single tube. You can do it pre-staining or post-staining. You can choose whatever you would like to do. But hence, because we wanted to do live barcoding, the Palladium-based barcoding was assuming that you were fixing and permeabilizing your cells before doing your staining. We have now in 2020, we released the CD45 cadmium-based barcoding. So we have 7 different cadmium label antibodies that would just pull in one single, that we would just like mix in the 7-choose-3 format, which allows you to do 35 samples in one single tube. So we can multiplex 35 different samples in one single tube, do the staining, and then run them all together. Then there will be a way to debarcode that, and we will be able to analyze them all individually. But if you're not happy with it, because CD45 is for immune cells, you can do your own barcoding. You can just like do your own barcoding for your target. So I will show you an example of that. This is an example of a cell type of a paper that was published last year in 2020, where they were able to do these morphometric maps, where they were able to actually, in samples coming from AML or ILL or different kind of like lymphomas, they were able to go to a resolution up to using targets against laminin, beta-actin, rRNA. So all these 11 different targets, they were able to create this morphometric map. And with that, they were able to also have like a distinguish which cell which, depending on the different expressions. But moreover, which comes back to the barcoding that I was explaining you earlier, what they were able to do is to actually barcode samples, to mix samples from different procedures. So for example, they were able to mix the samples from bone marrow, from blood, from tissues that were like from diseased patients compared to the healthy ones. And hence they were able to do this 52 parameter mass cytometry panel. So it's 52 markers in one single tube that will be able to run in the Heliosystem, do the high dimensional analysis and compare it to flow cytometry and macroscopy. So with that, they were able to classify the samples and create this morphometric map, which was stable all over the different type of samples, not only like healthy, but also disease samples. And they were able to compare it with like the macroscopy signals, which was actually easily comparable. So this will save you time and money. Another example I wanted to give you, so we have our own catalog. We have more than 900 antibodies already conjugated to metals. If we do not have the antibodies that you were looking for, we have this metal labeling kits, which work nicely with purified antibodies that are only in PBS. And then they can be customized, all the panels that can be customized by adding extra antibodies in the open slots. We have all these panels that are already like present in our catalog. And we also have some that are for mouse. I want to talk to you about this immune monitoring map panel, which is this MaxPro Direct immunoprofiling assay. It's a tube that contains 30 unique markers, which are in a dry format. So they are lyophilized tubes. Well, the only thing you have to do there is just to add your cells on top of it. So it's validated for PBMCs and blood, but also being used for other type of tissues. You add your PBMCs on top, you do your staining, and then we have a program that can, in 5 minutes, analyze automatically all your populations and identify 37 different populations. So this actually is super easy to implement. You do your staining, which is 4 or 5 hours, taking into account that you do PBMC isolation. If you don't do it, it's much faster. You do the acquisition 20 minutes per sample, and then you generate in 5 minutes. You are able to generate your report using the PathSetter program. What does the PathSetter do? It actually cleans the samples from dead cells, from debris, from doublets, and it allows you to generate this FCS file that will contain this perfect, beautiful, clean data. What will happen is that it creates this sensing maps, which is very similar to a TSNE map, where we're going to have these different cell types classified depending on the expression levels of the markers, and it's going to do that with a B-cell classification. Then you will have this report that will come out from this automated analysis, where it's going to have like this 37 different immune cell populations with the number of cells, the percentage of cells, also QC metrics, and it's going to be able, you're going to be able to export it, to print it, to do whatever you would like with it. So just for you to know, those are all the metals. These are the empty slots that we have. Those are the ones that are already dried in the tube. We have 14 extra channels that where you can add your markers if you would like to. So this is just an example with just like filling up the whole system. So you will have in total 44 different markers in one single tube. But we do have our expansion panels where we'll add extra some markers that we are already like kind of like pre-designed. But again, it's totally manageable. You can do whatever you want. So this is just the example with the B cell expansion panel number 1, which is activation markers for monocytes and B cells. There's also the panels for the T cells expansion, which is again, activation, exhaustion, whatever you would like to do. So it's reliable because it has been proven from site to site, operator to operator, machine to machine reproducibility. So you will have samples that will come from one simple blood banner, run at different sites, run in triplicate by different machines. So it was done by 6 or 7 different machines. And then this was published in 2019. And actually what it found out at the end, I'm just gonna go through it. What it found out is that the intra-site reproducibility was lower than 8% on the CV percentage, meaning that it's highly reliable. And the inter-site reproducibility was around like 15% as a median. So to be honest, what I think this this kit actually allows you to do is also to freeze your sample. So this just made things easier during now the pandemic. So you can buy the kit, you do the staining, you have this ready to use tube, you have the staining done, you fix the samples, you freeze them on -80. Those samples are frozen up to 4 months, and they can be shipped to a lab facility, which will run in the Cytometry. So this is something that with flow cytometry is actually not easy to do, and actually it's not even implemented. The freezing the samples is actually something that is very specific for mass cytometry, and this just makes your life much easier. So I hope that I was able to explain you that this is a comprehensive method, that is also something that it's efficient, that it's reliable, it's fast because you can do all your results in less than 5 minutes, and it's flexible because you can add extra markers if you would like to. I'm just going to show you 2 examples on how it was used in clinical samples. So this is just the first NIH. So this is the NIH clinical trial, the impact clinical trial run at the from 10 different participating sites, as you can see here, that will like to identify key features of COVID-19 susceptibility and progression in more than 2,000 different participants. And they use the MDPAC because it has this proven stability, and because they were able to stain, to freeze, and to ship the samples and do this automatic unbiased gating. So as another example, this is not using COVID. It's the antidepressant trial that with the P2X7 antagonist that was run running at the UK. So this is a phase 2 trial with 142 participants. That is an interventional clinical trial where they use like samples at baseline and samples at 8 weeks post-treatment. So samples were actually sent, Cambridge just sent the MDPAC kits to Glasgow and Oxford where they were collecting the samples. They did the staining, they froze the samples, they shipped them at the Kroc Institute in Cambridge where they were now doing the acquisitions and the analysis using MDIPA. So as you can see here, something that it's really easy to implement and to use. Now I'm going to talk about for the last 10 minutes on the Hyperion imaging system. So as I explained you earlier, the system that we just put in front of the Helios, it can allow you to measure more than 35 markers in frozen sections and in paraffin sections using conventional immunohistochemistry protocols. It allows you to go for tissue architecture and also cellular morphology, but also subcellular-like resolution. And it's very, very powerful because, again, you can do 35 different markers in one tissue slide. So how does it work? Just for you to know, you have your panel that you will design together with your FIS, your local FIS. This is just an example. We also have some kits, like the immuno-oncology kit that will allow you to actually measure immune markers, but also structural markers. You will do your staining as usual, and then we're going to image. So how does it work for imaging? We just kind of have a laser that will go over the tissue and will just like a blade do this ablation. We'll just lift this 1 micrometer at each time, like regions, and this will be sent into the Helios, which will measure at the end everything. And we'll create this analysis. In the analysis, we're going to be these images that will be created and will be analyzed later on. So right now we have more than 100 systems and more than 90 publications in the past 3 years. As you can see here, we published them in high impact journals, such as Nature or JAM or Frontiers Immunology, whatever you like. And it's quite an exponential growth. So we expect them to have like even higher in 2021 and 2022. And just for you to know, I'm just going to explain you 2 different examples that I think might be interesting for you as a clinical environment. The first one was a paper that was published in 2020 in Nature, which used like 352 breast cancer patients, and they used like tissue imaging to generate this beautiful maps. So I'm just going to go to the next slide, which is a bit more summarized. So they used 352 breast cancer patients, and they also used much healthy biopsies. So they did a panel which implemented these markers, which I think they're a bit small for you, but they are markers that actually look at immune markers, cytokeratins, also structural markers such as thymidine, collagen, everything you would think of, Ki-67, so kind of markers for proliferation of tumor cells. They were able to do the ablation of all the different samples and do this high dimensional imaging. With that, you can actually do these cellular communities, you can do clusters, you can do phenographs, you can do whatever you would like to. And with that, you can classify your tumors. And that they were able to relate it to survival analysis because they had the clinical data from these patients. And what was happening here is that they were able to identify these novel single cell communities within every tumor microenvironment. And they were able to link that to this correlation with the divergent clinical outcomes, because there were some patients that were not ever actually reacting the same way to the data, to the therapy. So hence they were able to link it to these different cell communities that they observe on the tissue slides. Another example I would like to explain to you is the tumor profiler study, which is this integrated multi-omic functional tumor profiling. Basically, they use like quite a lot of different technologies, including mass cytometry and imaging mass cytometry. It's a collaborative study with the University of Zurich, the Hospital University in Zurich, the Hospital University in Basel, and ZIP and Roche. And what they did is that integrate all this data to produce this final report, and basically is going for personalized medicine to try to go into the fast diagnostic loop for patients that really need this fast diagnostic loop. So they use this on melanoma and carcinomas, on AML patients. And what they did is as you can see here, they use Cytometry because it's high throughput capability and low cost compared to all methods. So just for you, this is just one of the examples that they give in this paper. Is actually they took a patient that had this metastatic acral lentiginous melanoma that progressed with anti-PD-1 therapy. So when they went for the classical diagnostic, they showed that this was an immune tumor with PD-1 expression on 80% of the T cells, so cells that were quite activated, and 10% PD-L1 expression on tumor cells. And that tumor had like this low tumor mutational burden because they had like this kind of expression of like, they also had like loss of NF1 exons 24 to 29, ATM, APC, and basically it suggested that MEK inhibitors targeting NF1 and POP due to the ATM loss of function mutation. So basically the standard recommendations were that immunotherapy was not recommended because of this low tumor mutational burden, and that the target for a small molecule therapy. So basically the patient should undergo palliative chemotherapy. But using Cytometry, what happened is that they were able to classify the samples, and they were able to classify that there were 35% of the cells that were tumor cells, 40% mostly exhausted CD8 T cells, which had expression of CTL4 and PD-1, not only PD-1, but also CTL4. And this was identified as a sample with immune-excluded tumor, right? Because then they were able to go with imaging mass cytometry, and they identified this significant correlation between the CD8 positive PD-1 cells with the PD-L1 positive tumor cells. So showing that there was something happening there. And that this was identified as a melanoma population positive for markers such as vimentin, MIF, or TIRP1, indicating this transitional phenotype with mesenchymal features. So this is something that was not excluded before. Using signaling pathways. So with Cytometry, you can easily go with panels that have the signaling pathways there. We are able to do very easily phosphoprotein stainings. They were able to show that this tumor-specific signaling pathways identify this phospho H2X expression in a very small fraction of tumor cells and low phospho-ERK signaling, which suggested to the authors that the MAP kinase signaling pathway is not the main driver of tumorigenesis. So hence, the recommendation changed for this patient. The recommendation was to continue immunotherapy with the approved anti-PD-1, but also add on top anti-CTL4, together with this off-label CTK4 and 6 inhibitor, as they might increase this tumor immunogenicity and be useful in this immune-excluded tumors, such as this one. So hence, as you can see here, by using the combination of all of the different technologies, including Cytometry and imaging mass cytometry, they were able to actually go for a totally different kind of diagnostic recommendation, which might have been useful for this patient. So with that, I would like to thank you very much for your attention. I'm happy to take questions on our live question session and answer session. So just to remind you, my name is Gloria. I am the FIS for South Europe of Proteomics and Fluidigm. And if you would like to contact any of us, any of your FISs in Europe, this is our email is eu.support@fluidigm.com. And with that, I would like to thank you, and I'll see you in a bit for the Q&A.
Bye. Thank you very much.
So if we have any questions from the audience, please type them into the chat box. If you go away from the presentation and you suddenly think of something that you'd like to ask the team, please stop by our booth. We have representatives there who can talk to you about any questions you might have. As Gloria mentioned, you can also email us directly at eu.support@fluidigm.com. So that's eu.support@fluidigm.com, and we'll be able to answer any of your queries that way too. So any questions from the audience? Just give you another 30 seconds. Okay, well, hopefully we will see some of you in the booth. And if not, thank you for joining us today, and I hope you enjoy the rest of the conference. Thanks,
Thank you very much. Bye.